All right, here we go, and welcome to the Antigens and Antibodies Dry Lab. You are joining first period, so thank you, first period. And you see the lab that we are going to work on. I highly suggest that you split screen the activity in the Google Doc with the camera view here that you're seeing. So like I was just saying, is that I'm going to kind of walk you through this as if you're hearing some of this information for the first time today. The purpose of this lab is to visualize what's happening within the blood in terms of the antigens and the antibodies and how they would react with each other. So I'm going to kind of walk you through the procedures. You'll notice that I have in the PDF, I have crossed out a lot of the whole selecting these and cutting them out and labeling them. But the blood bags have already been made with our different types of blood. We have type B, type O, type A, and type AB. So basic background is remembering that within the blood you have the fluid portion, which is the plasma, and the cells, which contain the red blood cells, the white blood cells, and the platelets. We're going to focus here on these red blood cells because they do most of the work in the blood. They are the most abundant. They carry the oxygen to the tissue and back. They replace the carbon dioxide back to your lungs so that you are able to exhale it. So what each blood bag is representing is the entire whole blood. So inside of it, we have the red blood cells, and the shapes on the outside are representing the antigens. Remember, antigens are on the red blood cells. And then in the bag, it might be a little bit harder to see. Let's move these guys. If I take out the red blood cell, we have the antibodies. The antibodies are located in the plasma. So they're just kind of floating around freely, ready to attack anything that looks like it should not be there. Specifically for type B blood, right? B blood has A antigens. If you can see that, they are labeled A, but they are a different shape. So notice how these A antigens will not attach or clot, or what we say agglutinate, to our B red blood cells because they don't match. So the A antibodies will not clot with the B antigens. All right. So now it says my instructions. Make this blood type your first donor blood and write the blood type on line 1 of the transfusion worksheet. Record the antigens on the worksheet, if any. So we are now down to the transfusion worksheet portion where you've got that big data table in your document. And I've actually filled in the blood type for both the donor and the recipient. That way everybody's line 1 is the same. A donates to A, line 2, A donates to B, etc. So when we're able to compare our results, we'll see that we should have the same results per line, if that makes sense. All right, so our first is the instructions kind of explain everyone would have their own blood type bag. They would go find a donor and a recipient, and they would match those up. So, in fact, I should probably have another bag. So, just a second on that. And as you can see, that is why we are not doing that in this way, thanks to COVID. All right, so our first donor is type A. Now notice it says, find another student in the room with a different blood type to be the recipient of your blood. Donors remove only the red blood cell from the bag, but the recipient must remove all blood components. Lay the model blood components for both blood types on the counter, desk, or tabletop, all right? So if type A is our donor, we are going to remove only B red blood cell. And you can see that type A has A antigens in our triangle shape. And then our other type A, right, would remove the whole bag. There we go. So we see again the red blood cell, and we notice that type A has B antigens. Remember, that's opposite. So this is something that you might already know, remember, or maybe you need just a little reminder that they go by opposites. So if you have an A red blood cell, okay, because the antigens on the red blood cell determine what type it is. So we have A antigens on our type A blood, and in it we have B antibodies. And the purpose of the manipulatives and the shapes are to show that when we take our B antibodies with our donor A red blood cell, they do not match, they will not agglutinate, and they will not clot. So our recipient, being blood type A, has A antigens and B antibodies. The donor had A antigens. Okay. All right, so I'm going to keep my red blood cell donor out there, but I have to replace my recipient. And now we're going to go to line 2, which our recipient is type B blood, looking for the B bag. We're going to dump it all out. So we've got type B blood. Oh, near. I'm throwing things. All right, so in our type B blood, we have our B red blood cell. And what's going on in our type B blood is we have our type B red blood cell, and our A antigens are just floating along in the plasma. They're hanging out here, and boop, 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 boop. Here comes my donor red blood cell. And all of a sudden, my A antibodies are like, oh my gosh, I recognize you, ah! And they will then match up, and they will attach, and they will agglutinate, or they will, what we say, clot, all right? So we can see that that is not going to be a good donor blood for B, all right? And again, I know this seems very simplistic for some, but we're going to go through it. I'm going to go through them all. You might start to pick up on it and realize you can do it without seeing each one, and that's okay. All right, let me put B back. And line 3 is going to donate our A red blood cell to type AB. So we are going to take all of our AB. Uh-oh. Is the bag broken? Is it missing parts? What's going on? It only has a red blood cell. Okay. The answer is it's perfectly fine. Type AB blood does not have any antibodies in the plasma. Okay, so there's no antibodies floating around. So when type A blood goes to AB recipient, it's just fine. There's not even any antibodies to attack the A blood cell. In fact, that is why type AB, specifically AB positive, is called the universal recipient, because it can take any blood. There's no antibodies that are going to clot with it. So really, you could go through this entire data table, and anything where AB is a recipient, it's not going to clot. So again, notice what's special about the AB blood cell is that it is codominant, has both A antigens and B antigens on it, okay? So if you need to see that, to write it down, our donor type, type A blood, right, line 1, 2, 3, 4 has A antigens on it. We're on line 3 with AB being the recipient. So it has both A and B antigens and no antibodies. All right. This one's an easy cleanup. Just put that one back in there. And now we are on to type O blood. So line 4 has us donating to type O. And here we see we do have a lot of antibodies. Oh, that one's just white. Okay. All right, so notice our red blood cell is perfectly oval. There are no antigens on our type O. But the antibodies are abundant. There are tons of them. So we have both A and B antibodies. So I like to think of it as none and all being opposites. Okay, there are no antigens, but there are all the antibodies, A's and B's. So when we donate type A, the A antibodies are going to go, oh no, invader, attack, attack, and they will agglutinate, okay? So that is why type A blood cannot donate to type O. What other types cannot donate to type O? Let's see, AB, well that's even worse. The A's and the B's are going to attack the AB, so that's not an option. How about type B blood? Right. Also, not an option. The B antibodies will attack the B antigens. So type O can only receive type O blood. Okay. All right, so we've went through lines 1, 2, 3, 4. And as A being the donor and all of the others receiving. So if you're ready, you can kind of go on and predict which will and will not clot. If you want to stay with me, you're welcome to stay with me. If you're ready to go ahead and answer those discussion questions, you are welcome to do so. All right, so up next, we are going to have type B blood be the donor. Just a second to resituate here. Actually, I think I'm going to keep all these out. Okay. All right, so we've got, this type B is messed up. Hang on. Type B, okay, our donor, and then we're going to go back through all of them as well. So line 5B is donating to A, dumping out the bag. I'm going to just try to do this as fast as possible, so do it based on what I know. So type A, sorry to move these from camera view. There you go. Type A blood has B antibodies. All right. So when donor blood B comes around, our B antibodies will attach to the B antigens, and that will clot or agglutinate. So that's going to be a no-go. All right. So line 6, we have B donating to B. So type B blood with A antibodies. And donor blood B comes along, also with B antigens. The A antibodies will not attack the B red blood cell. Okay. So obviously B can donate to B. They have the same antigens and the antibodies will not attack. Okay. Up next, we are donating to AB. So I am on line 7. AB blood, that's it. Remember, it does not have any antibodies. I don't know what these guys are. Okay. So AB blood has A and B antigens, no antibodies. So when our B red blood cell comes along, nothing to attack type B with its B antigens. That is compatible. It will not clot. Now we have lastly type O blood. Remember, type O has both A and B antibodies. Has both A and B antibodies. It has no antigens. So when type B blood comes along, line 8, right, those B antibodies are going to attack the B antigens on the type B red blood cell, and they will clot. B cannot donate to O. All right, so that's all for B being the donor. Now we're going to look at type AB being the donor. So lines 9 through 12. As we donate to type A, as A antigens and B antibodies. So again, the key is that they are opposite. We have an A antigen, the opposite of A is B, so they have B antibodies. Okay. Type AB blood comes along, boop, boop, boop. I gotta animate it. I'm sure this is getting redundant, so have a little fun here. And then our B antibodies are going to attack the B antigens on both AB blood. So that's a no-go. So basically, if the donor blood has the opposite of that, it's not going to fly because we have the B antibodies. If there's a B antigen, it is going to clot and it cannot donate. Okay. Same thing goes with type B blood. Type B blood has our A antigens. So when AB tries to donate, those A antigens are going to attack. Really, type AB blood is not a good donor blood unless you give it to another type AB, okay, because AB is the universal recipient. So otherwise, AB we're going to find is only compatible with another type AB, because remember, no antigens, so no matter what comes along, there's nothing to attack. Line number 11. Both of them have A and B antigens, no antibodies. It will not clot. All right, line 12, you should figure this one out by now, right? If we have type O blood, it's going to have our A and B antibodies. So when our AB blood comes along, it is most definitely going to clot, all right? And I know it seems redundant, but the fact that we're doing it over and over again is the point, right? You do it so many times, you're like, I get it, I finally get it. So hopefully that's where you're at. All right, lastly, lines 13 through 16. O is going to be our donor blood. So this one's exciting. We are going to finally see why O is our universal donor. So we're going to start with type A blood and its B antibodies. All right, and here along comes O. And look, there's nothing. There are no antigens on type O. So even if those antibodies tried, there's nothing to attach to. So it's perfectly fine. O can donate to A. There is no clotting. See that also with B. B red blood cell, B antigens, A antibodies. Line 14, O comes along to donate. Nothing to attack. It does not clot. Okay. Type AB with no antigens, or I'm sorry, no antibodies. AB has both A and B. O has no antigens, no antibodies, no antigens on O. So they are completely compatible. O is the universal donor. It will give to all blood types. And lastly, I need myself another O. Red blood cell. Looking through my pile. Here's one. All right, so I've got my O red blood cell, my A and my B antibodies in my type O, okay? And when my donor blood comes along, even though type O blood has all the antibodies in its plasma, the O donor blood doesn't have any antigens to it for it to react with. So lastly, line 16 will not clot. All right, so if you've walked through all 16 transfusions with me, you should have your data table filled out. Now it's time for you to answer your discussion questions.